Uh, Good morning, good afternoon, good evening to anybody, regardless of where you're from. My name is Bruce Yeager with the Society for Functional Precision Medicine. I'm going to go through a couple of different housekeeping items, and then we'll get into our October presentation. So first and foremost, like we start off with most of these activities, let's talk about membership. It's very easy to become a membership. You can join on our website if you're someone here for the first time or know somebody that you want to recommend. And if you haven't paid your dues for this year, please feel free to log on and pay your dues. Second thing is there was a notification was sent out to current members via email yesterday with our friend and colleague Tony Latai moving to the National Cancer Institute. Necessitates a couple different movements on the board, and so we've communicated that we are taking online nominations through the 17th for 3 different board positions, president, secretary, and a board member at large. So if you're interested or want to nominate somebody, please refer to the email that we sent out. There's an online link to the registration form. We need to have those in by 10/17, October 17th. From that point in time, we'll look at collecting all the nomination forms and then putting out communications on election activities with the people that have been nominated for self-nominated or nominated by others for board positions. So those are the 2 housekeeping items. And then now moving into what we're here to learn and listen to. So the talk for today is Functional Precision Oncology for Metastatic Colorectal Cancer. Our presenter is Kushtrim Kryeziu, and by training, he's a molecular cell biologist, but currently works at the Oslo Hospital University, and he's responsible for establishing their patient-derived organoid model program. Specifically, what he's going to talk to us about is his work in colorectal cancer today. But in his short career, he's been recently recognized for a couple different things that I thought were remarkable. In 2023, the Early Career Award from Oslo University Hospital, and in 2022, the Researcher of the Year Award from the Institute of Cancer Research. So again, he currently heads the Functional Precision Oncology team at the Department of Molecular Oncology at Oslo University Hospital, where he's focusing on developing clinically relevant preclinical models to advance individualized cancer therapy. And with that, I'll turn things over to Kushtrim. Kushtrim, the floor is yours.
Thank you. Thank you very much for the kind introduction. It's a great pleasure to be here. And I would like also to thank Tony for inviting me to be part of the society and also to give this presentation today. And I also want to congratulate him for his new role as the director of the NCI. Today, I will talk about functional precision oncology for metastatic colorectal cancer. But before I start, I would like to introduce the team. Since this is a team effort with people coming from multiple disciplines, surgeons, cell biologists, computational scientists, oncologists, and so forth. And this all started with Ragnhild Lute, professor in genetics, and Arild Nesbakken, a professor in surgeries, a GI surgeon, where they established the translational research program in colorectal cancer more than a decade ago. And this work is a product of that program. Also here, I would also like to thank Anita Svein for the large-scale data analysis that I will show you through this presentation. Colorectal cancer is a major burden in terms of mortality and morbidity. Every 35 seconds, according to data from the Globocan, a person dies of colorectal cancer. It has a relatively high incidence and mortality, especially in the developed world. And here you see indicated Norway and Denmark having as the highest incidence worldwide. Most of the mortalities are attributed to the advanced stage disease. 25% of the patients already present at the first time diagnosis with metastatic disease, and 20 to 30% of the other patients will develop metastatic disease during their disease course. Liver is the most commonly affected organ for colorectal cancer spreading. and 5-year relative survival rate is approximately 15%. In terms of biomarkers, there are not so many biomarkers as compared to the other major cancer types. RAS mutations, NRAS and KRAS are used to guide anti-EGFR treatment for those patients, so it's a contraindications. And here you see a group of biomarkers that are cancer agnostic, so tissue cancer agnostic, used for other cancer types as well, like MSI for immune checkpoint inhibition therapy. BRAF, that I will come back to in this presentation, is a little bit different in colorectal cancer as compared to the other cancer types. ERBB2 amplification that predicts response to ERBB2 inhibitors and fusion proteins. But overall, those biomarkers are not that frequent in colorectal cancer, with a total of less than 15%. But also the response rate, even if you have the biomarker, it's not very good. So there is still improvement to be done in this space. So what we do is we implement functional precision oncology, where we test for drugs, and that is known for this audience here. And we take into account also tumor heterogeneity, because we know from molecular analysis that colorectal cancer is a heterogeneous disease. During the presentation, I will show you a clinical study, where we use the sensitivities of 23 drugs to nominate the treatment for those patients, but also other drugs and experimental and off-label drugs that we think are interesting in some subgroups of colorectal cancer. So first, evidence trials. Evidence stands for ex vivo drug sensitivity testing of metastatic colorectal cancer, which is an off-label, open-label, single-arm phase 2 intervention trial. So there, after the patients sign the informed consent to participate, so we try to sample multiple tumors from different sites in the liver, that they are separated from each other. We grow PDOs from those and screen with a panel of 73 drugs. As I mentioned before, 25 of those are directly used to treat the patients back, so if we find a promising sensitivity. At the same time, so the tumors and the PDOs are regularly analyzed for mutations with gene panel sequencing. And then depending on drug sensitivities and DNA mutations, we can perform also other analyses, such as RNA sequencing or immunohistochemistry, to confirm some of the well-established biomarkers. And this data then is discussed in the molecular tumor board that nominates the treatment for those patients participating in the study. And the nomination criteria is relative drug sensitivity to the reference, which is depicted here. So in red are the two dose response curves of the respective patient as compared to the reference, which are all the gray lines here. And if this stands out, so we score this as a hit. And in addition, we take also the interlesion heterogeneity into account if the sensitivity is similar or different. Also important to mention that Evident is positioned at the third line or the fourth line, so after the patient have exhausted 2 prior lines of treatment. And we know also that the response rates for the standard of care decrease significantly. And in the third line and the fourth is less than 6%. So Evident is trying to improve this figure. Primary objectives of the trial are number of patients receiving a pharmacogenetic profile, guiding molecular tumor board nominated therapy, and the overall response rate of those therapies that are non-standard of care for metastatic colorectal cancer. And secondary objectives are progression-free survival, duration of response, overall survival, safety, and also the progression-free survival as compared to the previous line of treatment. So those are the preliminary data from the EVIDENT trial. So EVIDENT is still ongoing. So far we have screened 152 PDOs from 106 patients. So we have 36% of the patients modeled with multiple PDOs and 17% of those with longitudinal PDOs, disease recurrence or after surgery, or for example, progression to specific types of chemotherapy that they have received. And now the cohort can be divided into 3 groups. So 1/3 is showing resistance to many drugs, or they are not eligible for experimental treatment due to frailty, or they died during follow-up. 1/3 of the patients show sensitivity to standard of care, and 1/3 show sensitivity to off-label drugs where EVIDENT is relevant. So far we have 34 patients eligible for experimental treatment, and 8 of those patients have started or are still having the treatment. And here you see the targets that we have drugs, those patients are treated with. And we have 2 chemotherapies, a conventional chemotherapy, gemcitabine and methotrexate. 24 patients are still under follow-up. When they, for example, exhaust 2 lines of treatment, then we have a nomination ready for those patients. The time from sampling to drug sensitivity results is at the moment 7 weeks. So there are several advantages with our trial, I think, because we only analyze patients with metastatic disease to the liver, and we have a very large biobank of organoids that we can compare the sensitivities and the molecular markers to. And we use both drug sensitivity testing for most of the drugs that don't have a predictive genomic biomarker, but also complement with molecular profiling for biomarkers that are well established. For example, MSI for immunotherapy and gene fusions for respective treatment. And also we have the intrapatient interlesion heterogeneity that we take into account. And I will go through all those. So the large biobank, what I mean by that is so far we have 215 patients modeled with patient-derived organoids, with 410 of those, and this number is still increasing. So the more than half of them, 107 patients, have been modeled with multiple PDOs, where we have more than 1 tumor lesion modeled with the avatars. And 10% of those are modeled with longitudinal PDOs. So here you see a subset of 213 PDOs that have been characterized with different data levels, like the gene panel mutation, gene expression, biomarkers or diagnostic biomarkers like CDX2, keratin 20, and Ki67 here. So we have also 10 more proteins that we have tested. We have also characterized their morphology that I will come back to, growth speed, and of course, drug sensitivity. As compared to the other cohorts in metastatic colorectal cancer, our study shows that the top most mutated genes, the 10 genes, are not different. So we have in our biobanks models with mutated genes from all those genes here. And we have also different, for example, hotspot sites in the KRAS, NRAS, and the BRAF. They also reflect the transcriptome of the original liver metastasis. So as shown in here, so you see a very good correlation of the gene expression And here is indicated that the cell intrinsic signatures are very well correlated between the PDOs and colorectal liver metastasis. At the immunohistochemistry level, so an experienced pathologist has looked at the traits between PDOs and liver metastasis and concluded that in more than 80% of the sample pairs, so the traits are retained. Now, looking at the morphology of the PDOs, we think that there is some information there. We have identified two major types, so the cystic and the solid type. The cystic organoids have a well-defined lumen of the structures and an epithelial layer that surrounds the organoids. The solid is the opposite, so they are more chaotic in structure. And then we have also in-between structures so that they have both traits. What we see is that there is an association of the diagnostic marker expression, CDX2, with morphologies, where in the cystic morphology, so we see more of the CDX2 expression, and in the solid, we see more of the cytokeratin 20, as shown here. Those morphologies are also associated with gene mutations, in this case, RAS and BRAF V600E hotspot mutation, with the solids more being mutated for those genes. But also in the transcriptomics, so here depicted is cell intrinsic consensus molecular subtypes of colorectal cancer. So you see that the canonical subtype is more enriched in the CMS2. Importantly is also that morphology is associated with drug sensitivities. And in the first plot here, you see the mean sensitivity of 24 drugs. And as expected, the cystic or the more differentiated are more sensitive in general to the drugs, whereas the solid that is poorly differentiated, it's more resistant. And that is the case also for 5-FU, which is the backbone for chemotherapy that is used as standard of care for colorectal cancer, and afatinib, for example, that is also a drug that indicates EGFR inhibition in this disease. Next to the well-known associations with RAS and BRAF and anti-EGFR inhibitors, so we have seen all of those in the living biobank. So we were able to see also other associations in this graph here. You see co-mutations in the RAS, BRAF, and TP53 that associate with resistance to multiple drugs here. And for example, a commutation in the RAS and PIK3CA is more sensitive as to, for example, wild type or either of the two genes. With FLIRI or FOLFIRI, so we call the FLIRI as the preclinical treatment, that is the 5-FU, SN38, and leucovorin combination. So this is relevant as this treatment regimen is standard of care for patients. We can also dissect, for example, which of the hotspot mutations can associate with some of the drugs. So in this case, we have insulin-like growth factor receptor 1 inhibitor that associates with, so the BRAF hotspot mutation at the V600E position associated with resistance to this drug. So I'm presenting here DSS, assuming that many people here understand that this stands for drug sensitivity scores. There is also an association with the transcriptomic. So again, with ICMS, so we see, for example, the non-canonical ICMS3 associates with sensitivity to gemcitabine and aurora kinase A, alisertib. And this is independent of the KRAS and BRAF mutation status. Interesting is also that PDOs can be predictive for the patients since we know about the survival of some of the patients. So we see, for example, a couple of drugs, their sensitivity associated with overall survival from time of surgery. In this case, it's atorvastatin. So high sensitivity in the lab would predict a better survival for the patient in the clinic. Now, moving on to heterogeneity. As expected, intrapatient spatial heterogeneity is different from interpatient heterogeneity. So comparing multiple lesions within a patient as compared to lesions from different patients. But there is no difference, for example, between intrapatient spatial and intrapatient temporal. So those patients that we have samples from recurrent disease. I will focus on the intrapatient spatial heterogeneity in the next diagram, so which is depicted here. So we have here 64 patients. The intrapatient heterogeneity in drug sensitivity score, as shown as pairwise Euclidean distance, for those patients that have two or more than two lesions modeled with PDOs. So ranked are based on the median of the pairwise Euclidean distance. And underneath here, you have the morphology, ICMS, and the mutation data. And if the box is square, so that means that the different lesions or different PDOs from that patients are heterogeneous. There seems to be an association of between morphology and drug sensitivity in terms of heterogeneity, but not with the ICMS or the mutations. It would be probably better to look at individual cases and see how the heterogeneity can impact, for example, the disease course of a patient. So let's focus, for example, here with the extreme case of this patient with 3 models that has a APC mutation heterogeneous between the lesions. So here you have dose response curves from 3 different lesions analyzed from this patient. So lesion 1 has TP53 and APC mutation, whereas the other 2 lesions have only TP53 mutation. So the red lines from T1, as you can see from the curves, is generally more resistant to many drugs. Only not the case for this XIAP inhibitor, which is an LCL 161 drug, which shows the opposite effect. When we look at the patient history, so we have modeled the PDOs after 2 lines of treatment have been evaluated for this patient. In the first line, the patient received FOLFOX, which is oxaliplatin-based treatment. And you can see here that the PDOs show a pronounced heterogeneity to this drug regimen. And the patient progressed to this type of treatment. But then when they switched to FOLFIRI, irinotecan-based treatment, plus the anti-EGFR, the patient responded very well with a partial response in all 3 lesions that we have modeled. And you can see that is reflected also ex vivo, where those 2 regimens that we have tested didn't show a pronounced heterogeneity. So now moving on to patients with prospective treatment. So in this case, we have a patient diagnosed with a KRAS mutated metastatic colorectal cancer, had multiple lines of treatment, so with FOLFOX and FOLFIRI and bevacizumab, and also the TAS-102 plus bevacizumab, which now is the standard of care in the 3rd line. After that, we got biopsies from 2 lesions. We grew them as PDOs, and you can see from the curve, as I showed you before, T1 and T2 from this patient stand out as compared to the reference from the other patients regarding Volfiri. And this is interesting because you can see from the disease course that the patient has received Volfiri before, experienced stable disease, but then progressed to this type of treatment. After that, the patient received immunotherapy, but progressed to that. And then, since all the treatment options have been exhausted, it was then decided to re-challenge the disease with Volfiri based on the ex vivo drug activity. And surprisingly, the patient responded with a partial response. As you can see here, the measurement from 3 lesions and the CT scans from one of the lesions that was measured. And that was a case where we don't have a biomarker, a molecular biomarker, just based on drug sensitivity testing. Here I will show in parallel 2 patients that have a ERBB2 amplification confirmed and have received a Volfiri with bevacizumab and experienced a mixed response to the treatment. For the first patients, we have grown 4 different PDOs from liver metastasis and 1 from the primary tumor from the second patient. So we have 3, so 2 from the initial liver surgery and 1 from the relapse. And you can see from the curves that in the first case, there are differences in drug sensitivities. So here there is a more heterogeneity as compared to the second example where there is more homogeneity. So the tumor board decided then to provide or to prescribe the dual treatment with trastuzumab and pertuzumab. At that time, this treatment regimen was not standard of care for these patients. And in the first example, where there was heterogeneity, the patient did not respond to the dual inhibition of ERBB2, whereas in the second example, the patient responded, became resectable, and until today, the patient has no evidence of disease. So this has been also shown at the biomarker level, since ERBB2 can be stained with antibodies. So we looked, for example, at different lesions, and you can see here, for example, that some parts of the tumor express it, but some do not. But we had also lesions that had very high expression of ERBB2, and that very well corresponded with sensitivity to lapatinib, which is a ERBB2 inhibitor. Whereas in the second example, all the tumors that we have analyzed for ERBB2 expression showed a high expression and responded similarly to lapatinib. Yeah, so what are the limitations? So the limitations are the success rate. Of course, we are not able to model all the patients. So we have overall a 70% success rate, but also the assay time is an issue. Those numbers are comparable to other functional precision oncology studies on metastatic colorectal cancer, with the OPTIC study recently published 2 weeks ago, having a 52% success rate from biopsies and 11 weeks from biopsy to the drug nomination. Longitudinal heterogeneity can be an issue because for many of the patients, we receive the samples at the time of surgery, and then we wait until if the disease comes back, and during this time, so changes can happen and also sensitivities can happen. Drug combinations with unknown toxicity profile is also an issue because we find sometimes drug combinations, but those are not possible to implement because of the safety issues if the drug combination has not been tried before. So PDO growth efficiency is not always a bad news. For example, if we are not able to grow PDOs from the lesions that we receive. Here you see that the success of establishing PDOs is associated with patient survival. So those that are more aggressive, we are able to establish, and they have a poorer overall survival as compared to those that failed in the lab. And this is partly explained by the response to the chemotherapy or treatment that they have received before we received the sample in the lab to establish PDOs. To improve on this, so recently we have established a rapid drug sensitivity screen where we are sampling the tumors, and on the same day we mince them and we perturb them with a panel of 17 drugs. And after 5 days, then after 4 days of drug incubation, we make a report ready. And this can have a lot of advantages. And one of those is possible use in earlier lines of treatment. And as of now, so we are working on a national study that this screen can be implemented, for example, to nominate irinotecan versus oxaliplatin-based standard of care in the first line and improve overall response rates there. The disadvantage of this rapid screen is limited biopsy material that does not allow us to do other analyses. So think of immunohistochemistry, for example, or protein analysis and so forth. So some preliminary data from the rapid screen. So you see here the concordance of drug sensitivities of PDOs and matched tumor tissues are shown here. So you have here technical replicas. You have PDOs coming from the same patient. You have tissues coming from the same patient. So those have a very high Pearson correlation. And then here is the comparison of the PDOs versus the tissues. And there's a little bit of a range, but all of those are significantly correlating. So far, we have 9 PDOs analyzed from 5 patients. And this is understandable because this is in a way comparing PDOs after some passages in the lab where we remove the tumor stroma, whereas the screening of the tissues contains also components of the stroma. From the drugs in this 17 drug panel, so we include all the standard of care that is used for metastatic colorectal cancer. And it is interesting to see that sometimes we can predict even the molecular markers that are used in the clinic solely based on drug sensitivity data. So in this example, just showing you the dose response curves for encorafenib, so the BRAF inhibitor that doesn't show any activity, and the afatinib also doesn't show very good activity, but the combination of the two shows a clear synergistic activity, which indicates that this sample that we have tested is a BRAF mutated sample that was confirmed afterwards. So when it comes to BRAF V600E mutated colorectal cancer is the subgroup of patients with the poorest prognosis. So here, for example, we start from the biomarker. So we have 8% of the patients that have a BRAF mutation. Around 4% have a hotspot V600E mutation. And around 1%, 1.3%, for example, have a BRAF and the non-APC mutation. So the rest is APC mutated. There is a study that has indicated that a mutation in RNF43, for example, can serve as a predictive biomarker for the combination treatment with encorafenib and cetuximab for metastatic colorectal cancer patients. So those two patients with non-APC have been identified from the biobank only based on sensitivity to porcupine inhibitor, this drug. As you can see here, so the 3 PDOs from 2 patients uh, only respond to this treatment, whereas the other patients do not respond, which is in gray here. So in the gene panel, so we don't have, for example, those 2 genes that we sequence regularly, but this drug indicated that there is something going on at the RNF43 or this or R-spondin fusion oncoprotein. So we did then additional analysis. In this case, it was RNA sequencing, where we confirmed the RNF43 and this fusion protein with R-spondin 3. So we have then molecularly with a mechanism of action showed that the BRAF V600E commutated with RNF43 are more sensitive to the combination treatment that supports the data that has been published here with apoptotic cell death, whereas the other models show mostly cell cycle arrest, but not apoptosis. So in comparison here, 2 examples, one BRAF co-mutated with RNF43 and the other one with APC. So we see for all the BRAF V600E mutated samples, if we combine BRAF inhibitor with EGFR inhibitor, so the activity is always synergistic. A little bit more in the RNF43 mutated sample, and here you see a time-dependent measurement of the single treatments and the combination for those 2 models. And you can see that in the RNF43 commutated sample, the combination is stronger as compared to APC BRAF commutated. And adding chemotherapy in addition to that, so with 5-FU, so we don't see a difference, for example, in the RNF43 commutated to the BRAF, but we see a significant difference in the APC BRAF commutated example. And this is in line with a recent Breakwater phase 3 trial, where they have seen that combination of encorafenib and cetuximab can be improved if combined with conventional chemotherapy. As I showed you before, also in the patient population, we know that APC co-mutated to the BRAF is the majority of the patient, but the RNF43 mutation could perhaps stratify this group of patients that do not require the chemotherapy because that comes with side effects. So the other example starts with the sensitivity from the drug. So we don't initially know of the molecular mechanisms that, in this case, make the sample PARP inhibitor sensitive. So 7% of those patients have PDOs in this case that are sensitive to PARP inhibitors. Then we have looked at the molecular markers, so we know that TP53 wild type status is associated with sensitivity to PARP inhibitors, which is indicated here. We know that some of the patients have also homologous repair deficiency with mutations in the BRCA genes and ATM, but this is not in all of the patients. And we also are trying to improve the efficacy of PARP inhibitors because single drugs alone probably are not going to have a good success rate. So in those 2 graphs, you see PARP inhibitors as combined with standard of care, which is 5-FU and leucovorin in this case, which is significantly improved. And in another example, it's a more experimental. So PARP inhibitors and ATR inhibitors are even more active if provided together. So this triangle dot comes from a sample that has a BRCA2 and ATM mutation. So those samples are the most sensitive, as expected. Very good cohort of patients we have is with longitudinal sampling, so 22 patients where we have 80 PDOs that were established from samples resected after disease recurrence or progression to specific types of treatment, including patients in evident, for example, who have received experimental treatment. So we have before and after that treatment. And here it would be interesting to study the heterogeneity of sensitivities over time. So we know in the clinic that with increasing numbers of treatment lines, the sensitivities go down overall. But in the last example after this slide, I will show you that some drugs might show increased activity over time. But also it would be interesting to study the clonal evolution of disease progression. So how the different lesions, for example, differ in terms of gene expression or mutations. So this is the last example that incorporates all the data levels and analysis that I have presented so far. So this patient, 64-year-old male, also diagnosed with a KRAS mutated colorectal cancer, has been treated with Flox in the first line and responded very well to that treatment. So we received one of those lesions, grew the PDOs, and then did the immunohistochemistry for the diagnostic markers. We looked at the morphology as well. So you can see that the morphology changes from cystic to a more solid, mixed and solid state with increasing lines of treatment. So after this surgery for a long time, then there was no disease detected. And then when the disease came back, so they gave the same treatment that the patient responded in the beginning. And this is interesting because we have, for example, drug sensitivity data from this time point, and we see that the patient was sensitive at this time point, even after finishing the treatment, and could predict that in the next stage, the patient will still be sensitive to Folfox or Flox in this case. And that was indeed the case. So the patient responded to Folfox, and then you see in the second surgery and the third surgery at disease recurrence, the Flox treatment or sensitivity decreased. It was interesting to see that from all the drugs that we have tested, in this case, there were 47 drugs for this patient. So we saw a drug that I mentioned also before, the inhibitor of apoptosis protein inhibitor drug, LCL-116, showed an increased sensitivity in the PDOs from the recurrent disease. We looked then molecularly and found out that this drug effectively inhibits the IAP proteins, in this case, IAP1, XIAP, and cervivin very efficiently, and also was able to induce apoptosis in the PDOs from the recurrent disease, but not in the PDOs from the first surgery. Yeah, with this, I would like to end and give you only some messages from the entire presentation. So I presented the evident trial where we use the multimodal experimental diagnostics to guide in the third and the fourth line treatment, where we use both drug sensitivity testing and molecular profiling. We look at the heterogeneity when possible. If we have, for example, multiple lesions and we model PDOs of those, we take that into account, that we think it is more individualized drug nomination. And also I showed you that we have a large biobank that enables us to discover single drug and drug combination sensitivities in smaller patient subgroups. So with this, I would like to acknowledge again the two core groups from Ragnhild Lute in functional precision oncology and Anita Sveen's group in computational oncology that does the drug sensitivity analysis and large data analysis. And you have here again people from surgery, oncology, radiology, and pathology that contributed to the study. I also want to mention that with Professor Hegge we tried to implement some of the functional precision oncology to the clinic. So in the national infrastructure, which is called IMPRET here for precision cancer diagnostics. Also, I would like to thank the collaborators and funding and all of you for listening. So I would like to take any questions now.
Kushtrim, thanks. Hey, quick question. Originally, what was the turnaround time, for PDO development? And you said it sounds like it's substantially lower now with the rapid test. That's what's being incorporated today.
Yes. So for the PDOs, when we grow PDOs and amplify the material, we do all the analysis. So for research and diagnostics is 7 weeks at the moment. This is median. So we have from 2 weeks to a very long time. For the rapid test is 5 days. So it's 4 days, the drug incubation time, and 1 day for the analysis.
Okay. I think Mike had a question.
No, sorry, I was applauding.
Oh, thank you.
Thank you.
Anybody else on the call? How many have questions or comments?
Hello, this is Chris from Seattle.
Hi.
That was mind-blowing. That was really awesome. Congratulate you and your whole team and your
Thank you.
patients and your doctors. That was really inspiring. Thank you so much. Just one question. If you see, for example, examples of sensitivity to experimental drugs or drugs FDA approved for other tumor types, what's been your experience with in Oslo with being able to get those drugs to your patients, and who pays for that? Because here in the United States, it's extraordinarily difficult and it's extraordinarily frustrating. And most times we just can't get the drugs or we can't get the money to pay for them. That's what the oncologists have been telling us. So, yeah, that's one.
Yes. So for the evidence, so we have a panel of 25 drugs that are off-label, that are not standard of care. And for those drugs, so we get funding from a national infrastructure, which is called the Matrix, that supports clinical trials nationwide. So we get, for example, per treatment. So if we find reliable evidence that that drug might work for that patient, so we get covered by the Matrix. But very rare cases, so if the drugs, for example, are experimental, so it is very hard to implement that into the patient if the toxicity profile is not known for that treatment, or if the drug is not marketed in Norway.
A related question, have you had any luck with convincing pharmaceutical companies to pursue clinical studies for drugs you see that are effective in colorectal but not approved? Have you tried that avenue?
Not yet. So we have focused now on the, as I showed on the BRAF, V600E mutated cancers, where the treatment is approved for that subgroup. So we just want to see, for example, to identify biomarkers that identify patients likely to respond to the combination treatment, because in the second line, only 25% of the patients with V600E mutation respond to the combination treatment. And in the second example is the PARP inhibitors. So we have not come to that stage yet that we initiate a clinical trial. So first we have to do the preclinical work to show that for example, in novel combinations, we are testing 44 combinations with PARP inhibitors. So we have a focused project on that. And we have identified some of the promising combinations, but we need to gather more data to convince the pharmaceutical company to start a trial in colorectal cancer.
Okay, thank you so much. Thank you.
All right. Anybody else with any questions or comments?
Bruce, there's one in the chat.
Can you go ahead and go through that then, please? Joanne?
Yeah, Rita Fior asked, maybe I missed it, but in the therapy test, RDSS, the clinical predictive study comparing with patient clinical response.
Yes,
And
I see the question in the chat.
sorry.
No, we have not compared the response to the same treatments that the patient have received into the clinic, but that is something that we can do. So we have the data, so we just need to go and look. So we have not done that so far, but important question.
I guess while we're waiting, I'll ask another question. So you went up pretty quickly. Did you see sensitivity to, for example, MEK inhibitors in RAS wild type tumors? And if so, what do you think is happening there?
No, we have not seen that. So I have done the analysis, grouping different RAS mutations. So we see some sensitivity in the NRAS Q61 position mutation. But those mutations are not very frequent, so the numbers are not that large to perform a reliable statistical test. But we see individual cases that are very sensitive to MEK inhibitors.
And they can be, they're always associated with a RAS mutation or sometimes not?
No, sometimes not, sometimes not. So we can pinpoint some of the cases that are sensitive, but if we group all the RAS mutations together, we don't see a significant difference with the wild types.
Okay, well, that's interesting. Okay, great, thanks.
I have a question too. Maybe I missed it, but most of your PDOs, I mean, you obtain samples locally, right? From the Oslo, no? From the Oslo area, correct?
Yes, so mostly it's from the Oslo University Hospital, but we have received samples also from other sites of the country.
And how do you because this is something that we have discussed several times. How do you manage the transportation of the primary samples? Is there a system that you use, or have you tested a particular way that would make the tissue healthy for your platform? Have you tried generating PDOs, for example, from other countries?
No, other countries we have not tried, but we are, for example, here located not in the same campus where the liver surgeries are happening. So we have set up a logistics program. There is an internal bus that immediately after the surgery, so the sample is taken, is put into the transportation media and is sent here, so within 24 hours. So we have tested, for example, keeping the sample in the fridge in the transportation media for 48 hours. So we have not seen in efficiency of growth between 24 and 48 hours. We have also tested different types of media. And there we have also not seen a difference in the efficiency of growth. I think the efficiency of growth is mostly dependent on the viable tissue that we get from sampling. So that is probably the most important part to consider. If there is a pathologist that with confidence, for example, marks an area of the fresh tissue to be sampled that has viable tissue, I think that is the most important part.
So basically, in your experience, sorry for insisting on this, but I know it's an important question for this audience. So what's the maximum time that you would think that you could generate these models, and yeah, and which are in general the conditions? I understand the transportation, but that means that it's pretty local.
Yes. I can say so we have not done in many cases, but also we tried 3 days keeping the sample in the fridge, and we still were able to grow videos from those. But not many patients we have tried 3 days. Just to say reliably that, for example, also 3 days would work similarly as 24 hours. But I know that we have cases that were stored 3 days in the fridge, and then we were able to grow the videos.
Okay. Oh, thank you.
Okay, well, if we're out of questions, Kushtrim, thank you very much for sharing the work that you've been the work that you and others have been compiling over the past number of different years. Very interesting work.
Thank you. Thank you for listening.
Thanks everybody for attending this month's webinar. Look forward to seeing you a month from now again. Nominations, if you know somebody or want to nominate yourself for a board seat, needs to be to us by 10/17. Use the form in the link of the email. And then talk to other people about getting them involved with SFPM.
Thanks, Bruce.
Thanks, everybody. Have a good day.
Thank you. Bye.
Thank
Have a
you.
good
Bye
one.
bye.